To survive in a dynamic world, the prediction is important to compensate for the inevitable delay in neuron system. As flash lag effect shows, we perceive it as if the flashed segment lag to the moving one, but actually they are spatially and temporally aligned. This deviation is the result of the inference for a predictable object. In PPC, some neurons could infer the target state even when it was invisible, depending on the context. This neuron responded to the target which inferred to be moving during full vision and occlusion block, and barely active in the blink block with stationary target. The prediction also exists in the effector state estimation to achieve accurate movement. LIP and PRR are generally regarded as an important sensorimotor interface. As shown in an effector choice task, the response to sensory cue was high and similar in both areas before cue off, and activity separated when the movement type was confirmed, with LIP cells differentially active for saccade and PRR cells was rich specific. Here we trained monkey to do the occluded interception task with eye fixation and recorded the neuron activity of LIP and PRR. The monkey initiated a trial by touching a central dot, then a target appeared at a random location of a circle, moving circularly with one of 5 angular velocities. After moving between 400 to 800 milliseconds, the target disappeared, but kept moving for next 500 milliseconds. Then the central dot deemed as a go cue. The monkey was allowed to intercept the target at anywhere it wanted within 700 milliseconds. The random cue period and fixed occlusion duration made the monkey could decide the reach direction after occlusion started. We are curious about during occlusion period whether LIP and PRR represent the current state of stimulus or predict consequence of upcoming movement, or whether it is necessary to infer the invisible trajectory when movement plan is confirmed. To address these questions, we try to distinguish the fire rate in specific epoch corresponding to stimulus or movement information. For convenience, we divided the circle into 8 zones. The tuning curve and preferred directions was acquired according to instantaneous target location and reach endpoint. If a neuron turned to movement, it would have a consistent movement tuning curves and PDs across 5 velocity conditions. Otherwise, these curves and PDs would show the dispersed pattern. Here, we adopted an index to quantify the consistency of PD. PDs were projected on the PD from the static control. The index was defined as a ratio of the sum of projection and the sum of original PD length. Therefore, the PDs are more consistent when the index close to 1. Hence, we can get the temporal pattern of PD consistency. For this neuron, the movement coding was predominant during occlusion period. We found 2 major subgroups. Here are example neurons from purely sensory subgroup which showed persistent activity. The PRR neurons peak around the go cue, whereas activity of LIP declined after cue on. The stimulus index maintained around 1 until the middle of occlusion period, indicates that both neurons was turned to target location even the target was invisible for a while. The sensorimotor neuron were dominant in both areas. The example neuron in LIP exhibited a stair-stepping decay pattern, while the activity of PRR was persistent. Although the temporal pattern was different, the modulation property was similar. The PD index showed a transition from stimulus to movement coding around the cue off, suggesting they were modulated by upcoming movement after cue off. We counted the number of neurons, which were significant to instantaneous stimulus location, movement direction, and motion velocity, respectively over time, and found the proportion of stimulus to neuron in LIP was larger than that in PRR during cue and early occlusion period. The stimulus modulation decreased later and slower in LIP. For movement-turned proportion, it was larger in PRR, suggesting that PRR mainly focused on the motor intention, while LIP more focused on the sensory processing. The difference between 2 areas was more obvious in temporal dynamic in population level when we checked with Demised PCA. In LIP, the components corresponding to instantaneous stimulus separated well during cue and early occlusion period. However, similar component in PRR covered less variance, implying that sensory information contributed less to population activity in PRR, and the internal representation of target did not sustain across whole trial. The evolution of movement component was similar in both areas. The component diverged just before occlusion period, and dispersity reached to maximal around goal Q, suggesting the prediction of upcoming movement might gradually clear started from late Q period. To summarize, during the occluded manual interception with eye fixation, the activity of LIP and PRR neurons can be modulated by both current sensory information and upcoming movement. Once it is sufficient to form a motor plan, most PRR neurons focus on movement, while the LIP neurons can both estimate instantaneous target state and plan movement. However, the target tracking coding was only lasted until early occlusion period, which may suggest that the explanation of target state is no longer needed when motor command is confirmed. Thank you for listening.